Hello, Assalamu alaikum. In this video, we will understand about the bone-derived neurotrophic factor. The bone-derived neurotrophic factor is known as the basically BDNF. So basically it is present in our region in brain, important for the development of the brain and the neuronal glial or vascular constituent of brain tissue. In this way, the brain will develop and memorize the memory and other things, the cognition as well as the inhibition of the Alzheimer disease, dementia, and brain fog and brain memory loss. It is brain tissue and the basically synaptogenesis, neuroprotection, and short-term memory and long-lasting synaptic interaction that influence the mechanism of memory and cognition. Let's understand about the all things through a different diagram. So this is the cell of the brain, which that is brain derived neurotrophic factor gene consists of 11 axon, and it's a different splicing to various tissue and are responsive to different stimuli like the stress and the learning behavior as well as the feeding behavior, a lot more things which are related to the bone derived brain derived neurotrophic factor. The neurotrophic family which also involve include the nerve growth factor and the neurotrophin 3 and neurotrophin 4 factor. There is the presynaptic neuron and postsynaptic neuron. This presynaptic neuron will synapse with the postsynaptic neuron, and the glutamate will bind will lead to long-term potentiation and short-term potentiation. In this way, the synaptogenesis is important. It is due to the brain-derived neurotrophic factor. This brain-derived neurotrophic factor will stimulate through growth hormone, and this basically is the testosterone also involved for the male for the learning and the behavior of the male, and the growth hormone and IGF-1 for the adult and IGF-2 for the embryonic stage, which that will lead to develop the brain due to the increase of the BDNF. This all factor as well as environmental stress, learning, trouble, challenge, and the understanding, interaction, and experience will lead to activate the gene epigenetic acetylation. This epigenetic acetylation will lead to transcribe that certain gene which that was silenced, and in this way again this will be activate the silence gene will lead to new memory and new experience will be occur. This growth hormone also target as well as the testosterone will lead to activate the masculine behavior. The mRNA will lead to synthesis of protein is known as translation. In this way, they go into the endoplasmic reticulum and the pre-pro BDNF will be formed. This pre-pro BDNF will lead to pre-region and the pro-domain will be supplies in the Golgi bodies. In this way, here is the intracellular proteases, that enzyme, the proteases, which that supplies the protein. And in this way, the splicing of the protein, the intracellular convertase enzyme and furin, which that is a protein which that contain the basically maturation of the pro BDNF as well as the mature BDNF. This intracellular convertase will lead to remain in the vesicle, the intracellular vesicle, and while the convert, but remain in the inside of the cell, but not exocytosis. Whenever the need will occur, the calcium influx will lead to exocytosis of the mature brain derived neurotrophic factor, and on the other hand, the pro brain derived neurotrophic factor. Both neurotrophic factor is involved for the signaling pathway for the synaptogenesis and the growth of the neuron and protection of the neuron in the brain. The metalloprotease is enzyme used to convert this mature brain-derived neurotrophic factor from pro-brain-derived neurotrophic factor in the extracellular cellular matrix. This can be a MMP2, 9, and plasmin. Let's understand about the whole picture and how it works. In the extracellular domain, the tyrosine kinase binding receptor, which that bind the mature BDNF, when bound with the mature BDNF, will insert into the plasma membrane. And in this way, the homodimerization will lead to JAK and STAT also. But this basically another, the G coupled protein receptor will activate, and the phosphorylation and the complex formation will occur. In this way, will lead to cascade of phosphorylation. This cascade of phosphorylation will activate the gene through CRUB transcription factor. Bind will lead to CRUB dependent transcription will occur. CRUB is the gene, a protein which that is activated and phosphorylase will lead to become a transcription factor. And it is also tyrosine kinase binding receptor, BDNF receptor, will bind with the mature BDNF, will lead to activate the MAP kinase and the PI3K and AKT pathway. On the other hand, the phospholipase C gamma will convert this PIP2 to PIP3 and diacylglycerol. This PI3 and AKT activation will lead to anti-apoptotic neuron, the neurodegeneration inhibition, modulation of the NAMDA receptor, dependent synaptic plasticity for long-term potentiation, regulation of protein synthesis and cytoskeleton development, enhancement of the dendritic growth and branching for the new learning and new outcomes as well as experiences. MAP kinase activation will lead to signaling cascade, regulation protein synthesis, activation of ERK, CRAB, related path, and activation of cytoskeleton protein synthesis in the brain neural cell, and the activation of the dendritic growth and branching, same, and the calcium influx. What is the meaning of calcium influx? When the calcium move inside will lead to exocytosis of the granule like glutamate and the dopamine for the normal cognition and the normal function of the brain. The GTPase also trigger will lead to stimulation of the actin and microtubule synthesis will lead to cytoskeleton same and the growth of neural fibers. The phospholipase C gamma, which that will lead to activation of the CAM kinase and PKC, which that increase of the diacylglycerol and calcium ion concentration and enhancement of synaptic plasticity. Let's understand about the further. So we will also understand about how to stimulate the brain-derived neurotrophic factor. So this will lead to cyclic AMP activation through PIP3 pathway, will lead to activate also enhance the calcium, will lead to exocytosis of some vesicle, and this is CRAB, which that is CRAB dependent transcription will lead to MAP kinase, and the PI3K and mTOR activation will lead to transcription and translation of the AMPA receptor and the calcium dependent AMPA receptor and the NAMDA receptor. This AMPA receptor, NAMDA receptor, and the calcium dependent AMPA receptor, which that is important for the neuroplasticity and learning, and in this way the new synapse will occur. In this area you can see the calcium cyclic AMP will activate the protein kinase A, will lead to degranulation of the glutamate for example, and the HCN channel which that activate lead to influx of the sodium calcium, and the tyrosine kinase B will also bind with the mature BDNF, and the pro BDNF will bind with the P75 neurotrophin receptor, P75 NTR or TNF type, which that will lead to neuroplasticity enhancement in the synapse And this is basically the mature bone derived neurotrophic factor trigger and boost due to the omega-3 fatty acid through diet, coffee, and the green tea. And the leafy greens basically containing magnesium for synthesis of the protein in the ribosome, and the resveratrol and cocoa, polyphenol and sulforaphane and olive.